Welcome to part 4 of the Breeding Value Predictions module of the TLR course at the University of Pretoria. In the first 3 parts, we basically looked at how the equations are constructed in terms of the drafting of matrices, and we also basically worked on a few examples. The first example we did as part of the course, and then in part 3 you were given the task to draft these matrices, do the matrix calculations, and come up with solutions. First, best linear unbiased estimates for the fixed effects, where there's corrections for the size of the groups as well as, or the fixed effect levels, as well as the an adjustment for the possible genetic differences amongst the animals in the groups, or the levels of the fixed effects. You also got solutions for the additive genetic merit or value for each of the animals that you used in the examples. In part 4, we are going to move over to the different types of models, so the complexity of different models in blood breeding value predictions. So you can follow with me through this, and although we're not going to work on solutions for each of these cases, the basic principles on how blood breeding values are derived will basically remain the same. We then kick off by looking at what I would like to call extended mixed models for breeding value predictions in farm animals. So the examples, like I said, are very basic examples where we had a limited number of fixed effects or levels of fixed effects, like sex of the animal, or maybe farm. In the other case, we looked at the different parities, lactation 1, 2, and 3, and we looked at 2 different growth tests for young bulls, where the example that you use for average daily gain, breeding values for average daily gain. So those were very basic models where we did not look at other more complex models in terms of fixed effects, but also additional random effects. So in all the examples we did up till now, we only used one random effect, namely the additive variance or the additive breeding values of the specific animals as a random effect. So let's then kick off with some models of importance in blood breeding value predictions. The examples we used up till now is basically a single trait animal model where a breeding value is derived for each animal. And you also saw that each animal has its own equation, and you also then get the solution for the additive variance. That's the unknown or the random effect. So the observations are on one character, and there were no other random effects. So the only random effect was the additive variance. Random. Why is it random? Because each animal gets a random sample from its parents, and we don't know beforehand which random sample it got from its parents. Was it the bad genes, or was it the good genes that the animal received? The other model of importance is the so-called sire model. Now, the sire model was the very first models when blood kicked off way back some years ago, and the sire model is based on the progeny records of sires. So in the dairy world, when blood kicked off, when Professor Charles Henderson started with blood, he started with a sire model, simply because of the fact that the computing facilities in the world were not adequate enough to handle more complex models. You basically work with the mean or average value of sire progeny groups, and the problem is also that there's no real correction for the differences between dams. So you have some biased, if you think about the U in blood, unbiased prediction. So you had some bias in terms of the differences of genetic levels between the dams. That was corrected later on with a maternal grandsire model, where also the maternal grandsire was included for each animal as an additional random effect, and that caused some sort of correction for the possible bias between dams. So if you had preferential matings, some size being used on better dams, for example, that type of bias was then in some sense nullified in the process. From the single trait or the animal model, the so-called RAM model, the reduced animal model, was developed, and that the big reason for that was that it reduced the computing power needed for the animal model. So you had, let's say, 1 million animals in your or with 1 million equations, obviously, plus the levels of the fixed effects. If you have another 1,000 fixed effects, you add another, your matrix grew by another 1,000 by 1,000, and the reduced animal model was just a mathematical way of dealing with these large matrices. So breeding values were only fitted for animals that have progeny records, so you excluded animals in the equations that was nonsensical, who didn't have measurements and did not become parents, and the breeding values were then derived for parents, and all the other breeding values, the estimated breeding values, were derived from these parents, plus the corrected phenotypes. The corrected phenotype then is the phenotype or the measurement of the animal within its own contemporary group. So that was basically just a way of dealing with these large matrices to assist one in the process. Good. Just some other models then. The next model of importance is a so-called repeated records model. I can just use one example. Let's use the example of repeated records in terms of a lactation, first, second, third, fourth lactation. One can either fit a multiple trait animal model, and we'll talk about the multiple trait model, or you can see the lactations as repeated measures of the same trait, repeated year after year. So, and you then need to include the phenotypic correlation between recordings, and that then would make out the repeatability in the model. So there's an additional random effect called the repeatability, and we also see that as we call a permanent environmental effect of each animal. So the animal itself becomes a random environmental effect from year to year or recording to recording. So that was then also an extension of our basic model. Then a very important model, and we'll talk a little bit more about this later, is the so-called maternal effects model. We know that, for example, in young animals, while they're suckling on their dams, beef cattle, for example, piglets on their dam, or lambs on the ewes, that early growth is to a large extent influenced not only by the additive genetic merit of that young animal, but also the additive genetic merit of the dam that suckles that calf or lamb or piglet. So that means that there's an additional random effect, the additive effect of the dam, that will influence the weaning weight of that young animal. So, as far as the young animal is concerned, the maternal effect is an environmental effect. But we also know that the maternal effect from the perspective of the dam is an additive genetic effect. So that is the extension then of our more simpler models to include, for those traits that are maternally influenced, we also include the dam's maternal effect. So for example, in weaner calves, we have repeated records in terms of the dam weaning a calf every year, but we also have the maternal effect per year or per weaning of a calf. So both these would then be included in an extensive model if we have to predict the breeding values for weaning weight in cattle, for example, or in sheep. A very good a big extension of the basic models for breeding value predictions with the development of the multi-trait model. We also know that all traits are somehow genetically correlated, so there's an additive covariance between all traits. It's either by means of the additive genes being shared for the same trait, or where they're situated on the chromosome close together. So you could have a loosely correlated um, correlation between two traits, or two traits can be very highly correlated. So in terms of the development of blood models, the multi-trait model was a big step forward. So we know that we could either use different or separate single trait blood runs, but that will give a false impression in terms of the breeding values for the different traits. But if we include them all in the same big blood evaluation, where we also include the covariances or the genetic correlations amongst those traits, it also assists us in an accuracy increase in the prediction of our breeding values. And this is especially true where we have sequential culling. That means that you start off with, let's say, 100 animals where you have birth weights. Some die before weaning, and you end up maybe with 95 animals at weaning. You cull another 20 animals. So at yearling weight, you have less and less animals, and also, let's say, at 18 months, you have less animals again. And by including the genetic correlation amongst these traits, birth weight, weaning weight, yearling weight, and 18 months old weight, you increase the accuracy of your prediction for the later weight traits. We then continue with the discussion of more complex models for blood breeding value predictions. The next one I would like to discuss is the so-called random regression model. This is basically where traits are expressed repeatedly over time and maybe in different environments. And these effects of the environment changes gradually along a specific trajectory of time. A very good example of a random regression model would be the lactation curve of a female while suckling, let's say a dairy cow. So we can actually mimic the lactation curve by fitting a so-called random regression model, where let's say we have 10 test days during a lactation of 300 days, and in each of these, the actual production of the cow would be influenced differently in terms of the environmental influences. Whether she's pregnant or not would have an influence. Days in milk will have an influence. Whether she was on heat at the time of the test will have an influence. And those are all random effects that you did not account for, and that can then be included in your random regression model. So it's basically a regression fitted, but that regression is influenced by some random effects, and that can then be included in your model. A few other aspects that you can include in the models are models accounting for heterogeneous variances. This is basically where the spread of the phenotypic values can vary between, let's say, different environmental groups. And here, for example, in a specific contemporary group, in an area, maybe in some areas in a country, or within a herd year or season, there can be huge variances in the spread of the phenotypic values that you record or measure. And this will have a marked influence on the stability of your breeding value predictions, and you can then include that as an additional random effect in your models. A very important other possible factor is a so-called genotype environmental interaction, and that goes hand in hand with heterogeneous variances, where you also include the possibility of a genotype by environmental interaction. For example, to account for the possibility of preferential treatment of the progeny of a specific bull or bulls, you can also include the bull by environment as an additional random effect in your model. So that would then account for the possibility of a genotype by environmental interaction. We could also include as an additional random effect a so-called temporary environmental effect, and this, for example, would be, let's say, as litter. The litter groups for piglets, it's just by chance that a piglet will land in a specific litter, so that will be seen as a temporary environmental effect, even in the cases where the manager of the pig farm will actually exchange piglets between litters, or let them suckle in a different dam, and that could then be included in your blood models. And then we'll also talk about that a little bit later as a so-called non-continuous or threshold expression of measurements, and that is where you have threshold traits or binary traits such as calving or not calving. It's expressed as a full calf or not a calf in terms of calving, but we know that there is a continuous scale of additive variance. Animals differ in terms of their ability to be able to calve irrespective of the environmental effects, or because of the environmental effects, but the expression is it's not a continuous expression. It's either a calf or not a calf. It's a binary expression. So that needs to be taken into consideration as well in some of the models. So let's just then start looking at the simple blub animal model. So our simple blub animal model, and this is also a little bit of a revision of what we have done up till now. We have records of individual animals within contemporary groups. This is very important. Remember that blub basically works with the deviation of each animal's recording from its contemporary group. The actual value or the actual measurement is not important. What is really important is the deviation from the mean of the contemporary group. So mostly these models will be defined based on biological knowledge and some statistical methods, how to decide which fixed effects would be included in your model. We know from our biological knowledge that males, for example, will weigh heavier than females. So we know that sex should be part of our models, but we can also then test statistically whether sex of the animal will have a meaningful effect on, let's say, a weight, for example. So based on the biological knowledge and also with some statistical tests will convince us or convince the user then of a model, whether that will be included in a model. So a simple model for single traits will have a single random effect, like I said, usually an additive, or what we also sometimes call a direct genetic effect. And we also assume that there's no genetic covariances with other traits. So this will be a simple single animal model where if we have 5 traits, we will run 5 blood runs each, and we then assume that there's no genetic correlation or covariance amongst those traits. So that will be a simple animal model. So just to give you maybe another or a glimpse from a different angle, if we have a specific bull within its own contemporary group, it was performing better than its peers. So these animals were all subjected to exactly the same environmental effect or fixed effect, and these then represents, these little X's here represent the performance of other animals in that contemporary group, and that specific bull, the black cross X here, is the sire's performance. So in a simple manner, we would say that the additive effect or the breeding value of this bull would be equal to the heritability multiplied by its own performance as a deviation from the mean of the group. So that will be a positive value because the animal is performing better than the mean. Let's say he's mated to a female, a cow, that in her contemporary group, she was performing a little bit under the mean or lower than the mean of the group, so she will have a negative breeding value. So in our first prediction of the progeny of these animals, our prediction would be the breeding value of the sire plus the breeding value of the dam divided by 2. So our prediction would be that their progeny would perform on the mean of a new contemporary group, let's say in the next year or whenever these animals were weighed. But if we have an individual animal as the progeny of this group, its own performance within its own contemporary group would then also contribute to each breeding value, plus then the expected breeding value based on the parent average. We call it the parent average or the mid-parent value of the sire of the dam. So this blub will first predict basically the average value or the mean value between the two parents, and then the deviation from that mean will then add it in our breeding value prediction. So we also see this then as a step up in the accuracy or reliability of our prediction based on the parent average plus the animal's deviation from its own contemporaries. So and obviously in each case the heritability is taken in consideration, as that is the additive variance relative to the total variance for that specific trait. So this can then be extended way further in a total population. So that is the part we were basically looking at, where we had the dam and the sire in terms of their contemporary groups, the animal's own breeding value in each contemporary group, and then it might have progeny, and the progeny would then each of its progeny's performance within their contemporary groups will then contribute to that specific animal's genetic merit or additive value or breeding value. The same is then true for all other relatives that are maybe further removed. Obviously this animal is 50% related to its sire and its dam, and also 50% related to each of its progeny, and that will all contribute to the breeding value of that specific animal. If we talk about 50%, it means that it is as if half of the animal will be tested in each of these environmental different environmental groups or fixed effect groups, geoseasons, or whatever the case might be. So just coming back to the difference between random and fixed effects. If we think about random effects are usually genetically based. It's usually additive genetic effects. It can be direct or indirect. Direct, for example, the direct growth value of the calf pre-wean. Indirect, that would be the additive effect or additive value of the mother, her effect on the growth of the calf. So it can be direct or indirect. It can also be a mixture of additive and fixed effects, like the inclusion of sire by herd in your models to account for the possibility of re-ranking over contemporary groups, or a so-called genetic by environmental interaction. So that can also be included in your models. We can also have some non-additive genetic effects like heterosis or a dominance effect. For example, where you have cross animals, that you have purebred animals and crossed animals in the same genetic evaluation. You can also include the percentage or the constitution of those animals, how they constituted, as a so-called heterosis or dominance effect in your models, to account for possible non-additive genetic effects that can cause differences in the performance of the animals. We also have other random effects like temporary effects, like litter effect. I've mentioned litter effect in pigs, and we also have some randomized permanent environmental effects. So the repeated measures, let's say you have a cow that weaned 5 calves in her life, but she's basically a permanent environmental effect for each calf over a time period, and so you can include that permanent environmental effect in your model as an additional random effect. The fixed effects, that's also, remember, P equals G plus E, so the fixed effects you can also then classify it as big E, or environmental effects. You get different types. You get classes of fixed effects, or direct fixed effects, and expressed as classes, and you also have continuous or covariates that you can fit to your model. So classes have a distinct and different levels, like sex, for example, male, female, so it's only 2 classes. You can have lactation number. Let's say you have 3, you consider the first 3 lactations, so you can have lactation 1, 2, and 3 as 3 classes. You can have two growth tests, so you'll have 2 growth tests as 2 classes. The example we used previously in this course. It can also be what we call concatenated, like herd, year, season. So we have a class for each herd, year, and season of birth of the calves. So if you have a herd with 2 seasons, you'll have herd 1, season 1, and season 2 as 2 different classes. Or year, 2 years, and you have 2 seasons in each of those years, you end up with herd 1, year 1, season 1, herd year 1, year 1, season 2, and so forth. So that allows for main effects and all the interactions that could possibly be between all these classes. Then in terms of continuous covariates that you can include as fixed effects or a regression, there's no distinct classes, and this can be linear or non-linear. An example of this would be the age of the animal. We know that the older the animal, the higher the weight or body weight of the animal will be. But it's a continuous difference. Each day that the animal had more to grow, the probability will be that it will be heavier. So you can't have distinct classes to say, let's, for example, say young, older, and eldest. You work on a continuous manner where you include days of age in your model. For example, also the age of the dam. We know that young females will wean lighter calves, for example, but it's on a continuous scale. It's non-linear because she will reach her peak at, let's say, 4 or 5 years, and then at 8, 9 years she will also produce less milk, and she will wean lighter calves. So you can fit that as a regression in your model, the age of the dam, for example. So this is an illustration of 2 fixed effects. We have the fixed effects herd, year, season, and we have a second fixed effect, sex. So in this specific herd, year, season, you will see that the male calves are heavier than the female calves, so the mean value of the male calves would be higher than the females. Most of the animals around the mean each time. On a different farm, maybe different herd, different year, or a different season, you will see the same pattern where the male calves would be heavier than the female calves. But all in all these animals, because they were born in a different herd or year or season, will be heavier than the first, let's say, the first farm, or this could be a better year. And we can continue along these lines. A different herd, maybe, again, you will have heavier calves generally, but the male calves will be heavier than the female calves. And this is obviously a very good environment where these animals are much heavier than this environment, but still there's a difference between the weight of the male calves and the female calves. So this just illustrates an example where we have to include both of these so-called fixed effects or environmental effects in our model, not just sex and not just herd, year, season. We have to include both of these effects in our blood model. This is an illustration of where we fit a continuous variate or a covariate as a fixed effect. I have mentioned this. If we were to predict the breeding value for 305 days milk production or lactation milk production, we know that young heifers will produce less milk, not because they're genetically inferior, but simply because they're younger. As they become older, they will produce more milk, irrespective of the genetic merit of the animal, and we have to account for that as an environmental effect. So we will fit this regression in our blood model. At maturity, she will be at a peak production, and that will remain for a few lactations, and then when the female becomes older, lactation production will drop again because of age, not because of genetic merit. So you can fit this then as a fixed effect, this regression in your blood model for, let's say, 305 days milk production or lactation milk production. The same obviously is true in terms of weaning weight, where the maternal effect on the calf suckling on the cow needs to be accounted for in terms of the age of the dam, and you can also fit a model like this or this effect in a model to account for the possible differences. We can also extend the animal model by including some additional random effects. We have mentioned some of them already. We can fit temporary or permanent environmental effects like litter or random permanent cow effect. We have mentioned the possibility a bigger litter means that each of the piglets will get less milk from the dam, not because the dam is genetically inferior for milk production, but simply because there are more piglets to feed. And that is a so-called temporary effect on each of those piglets because of the fact that they come from a bigger litter, for example. Also, the repeatability of the cow from one lactation to the next, or one calf to the next, one parity to the next, can be seen as a so-called permanent cow effect from one calf to the next. We can also add additive covariances, that's a genetic correlation for more traits, and that assists one in a case, for example, where you have sequential culling, where you start off with many animals for weaning weight, but you have less, because of culling reasons, you have less animals weighed at a later age, and by fitting the covariance or the genetic correlation between weaning weight, let's say, and yearling weight, your predictions on weaning weight will be more accurate, but especially on the later weights where you have less animals in and where the contemporary groups construction is different because you have less animals with weights in those contemporary groups at, let's say, year-old weight. So this accounts for non-recording. Also, sex-limited traits where you can't measure milk on a bull, but you can also include some of the traits on the bull in your covariance structure. I have mentioned sequential culling as well. And the size of your coefficient matrices then grows by 2 to the power of n. So if you have, you start off with a coefficient matrix of 1,000 rows by 1,000 columns. If you include 2 traits, it will be 2 to the power of 2 because of 2 traits, so it will grow by a factor 2 times 2, 4. So that your new coefficient matrix will not be 2,000 by 2,000, but a 4,000 by 4,000. So you can immediately see that there's a rapid increase in the size of your coefficient matrices if you include more traits in a multiple trait. Good. We can then look at the beef cattle example where we have extended our model. We have the weight of the calf here. We obviously know that the sire, the genetic relationship with the sire would be 50%. It also have a genetic or it's related to its dam by 50%, but we see an extra contribution of the dam. We have a maternal effect, and that's an additive effect of the dam, plus we have a permanent environmental effect. So from the dam side, we do not just have the additive genetic effect as a random effect, but also the maternal effect as a random effect and the permanent environmental effect as a random effect. So that is an extended model for a weight like weaning weight. Birth weight is also maternally influenced, weaning weight is maternally influenced, but yearling weight to a lesser extent. But this calf is also weighed within its contemporary group in all these different for all these different traits. So the deviation from its contemporary group plus the complex model with added random effects, maternal and permanent environment, plus the added additive genetic direct effects will then give you genetic merit values for this calf. So in effect, you will get two solutions for additive effects: the direct growth of the calf plus the maternal effect based on the dam's maternal influence on the weight of the calf. So in this case, where we have birth weight, weaning weight, and yearling weight, the biggest influences maternally would be on birth weight and weaning weight, because that's when the calf is very much dependent on the maternal effect of the dam. To a lesser extent at year-old weight, because the calf has to look after itself after weaning. So if we come back to maternal effects, the weaning weight of the calf is because of its own genetic merit and environmental influences. Its own genetic merit is the additive genetic merit for pre-wean growth, and there's some other non-additive. The camp it was kept in, the year it was born, is it a late season calf, is it an early season calf, and all that. So that would be some non-additive effects as well. Then also in terms of environmental effects, the dam is also an environmental effect for the calf, but the dam has a genetic merit, and there's some environmental influences on her ability to look after her calf. The genetic merit is then the additive and non-additive effects, additive that can be transferred to the next generation or our breeding values, non-additive such as dominance and epistasis effects. Just to come back to this non-additive, I just want to correct myself. This non-additive effects on the direct growth of the calf would also be maybe the effect of heterosis or dominance effects or epistasis effects. So it's not an environmental effect. I just want to correct myself on that. So coming back to the dam's own performance, she will have a genetic merit for to look after the calf, but there will also be environmental influence. It can be a contemporary group effect. The calf she has to raise this calf in the year and the farm, and maybe some other fixed effects. So just for the calf itself, other environmental effects would be its contemporary group, herd year season, and treatment if there's a treatment difference, and possible other fixed effects. So you can already see that in terms of maternally influenced traits, we have very complex blood models where the weaning weight of the calf is not simply because of the additive effect of the calf, its genetic merit, and some environmental influences. Within, as far as the calf is concerned, the environmental influences, you also have to add some additive effects, and that's the genetic merit of the cow to look after the calf. So just to illustrate what the maternal breeding value of a bull, although you cannot measure the maternal value in a bull, you can measure it in its related animals, female related animals. So if you have a specific bull with a direct breeding value of plus 10 kilograms, that's 10 kilograms heavier than the population, and a maternal value of plus 7 kilograms, what does it mean? It means that you will not be able to measure the maternal in its male progeny. You will only it will basically transfer half its direct, obviously maternal as well, to its sons, but you will not be able to measure it, and in the grand progeny half of that again. So that advantage, given that in each case these males were mated to average cows. In terms of maternal, you will still have half the direct from the bull, so in the first generation you will have exactly the same effect on its daughters, but in the next generation it will transfer half the maternal effect to its daughter, so that's half of 7, and that will be a 3.5 kilogram difference in the grand progeny from the female side. And that's basically what the maternal breeding value, or the additive maternal breeding value, is all about. In a pig example, just to show you a more complex model in a pig, obviously again half from the sire and the dam, we have the contemporary group deviation of each little piglet, but we have temporary environmental effects in terms of litter. So irrespective of, let's say this is back fat, this is weight at specific age, and this is the longissimus dorsi muscle area that was measured, but litter would then also be included. So you have your additive genetic effect, you have a maternal effect, you have a permanent environmental effect, but you can also have a temporary environmental effect for that piglet in the litter. This is a very complex model for pigs, for example, where a trait of interest is number of piglets born alive. I'm not going to go through this, but you will see that even the service sire will have an effect of on the number born alive, simply because it might have an effect of the number of oocytes that the sow will produce at serving due to the environmental effect of that specific service sire. So the service sire is also then included in your model. Remember, number of piglets born alive is a trait of the dam, of the sow, but the sire being, or the herd, the sire being used on at mating will influence phenotype as well. So there are also more complicated models like repeated measures. We have mentioned that, measured on the same animal, repeated from one day to the next. Test day records, for example, in dairy cattle is a very good example of such repeated measures. You can either work on test days, or you can also use a random regression model for that. And that's where we mimic, or we fit the lactation curve as a regression, and it's also possible for growth traits to fit the growth curve as a so-called random effect. Then there is also the possibility of so-called transformation. We have mentioned threshold traits. It's what I would like to sometimes call all or none traits. You either measure something or you don't. You either a cow calves or she doesn't calve, and that's so there's a specific threshold. Or traits that's not normally distributed, like somatic cell count, for example, in dairy cows. So you have these spikes or peaks. Let's say the average somatic cell count in the contemporary group is roughly, say, 200 cells per milligram. And if a cow attracts mastitis or have subclinical mastitis, that jumps to 1 million. So you have this very uneven distribution, and you have to make some transformations to enable you to select against mastitis in dairy cattle. So this is an example of a threshold trait where if this is the genetic merit of cows in terms of whether they will calve or not, you have a specific threshold. That could be a drought. So the threshold can move from year to year or season to season. If the threshold moves to the right, so this is a more difficult season, a specific cow with here where my pointer is, let's say she's on that point in terms of a genetic merit to calve or not to calve. In a good year she will calve, but if this threshold moves towards the right and she's just on the threshold, she will not calve because her genetic merit does not allow her to calve in a bad year. In a very good year where you have, let's say, a 99% calving, the threshold will be very close towards the left. So it's basically expressed as calved or not calved, but on the so-called underlying scale, there's genetic merit differences amongst our cows. So we have to, if we want to predict calving rate, let's say the daughters of a bull, we need to take that into consideration in our models as well. So this then brings us at the end of part 4. It was a longer lecture than the others. We basically talked about the extension of models from a very simple model where we only started with the deviations from a contemporary group to very complex models where we fitted additional random effects. We also looked at the different ways that we can fit fixed effects, some transformations needed where we do not have continuous variation or where we have an unequal distribution of our phenotypic values, like in the case of somatic cell count, and how we then change either our phenotypes or we include additional effects in the models to get to a situation where we have a clear view of the additive genetic differences amongst the animals or the breeding values of the animals. This then, as I said, concludes part 4 of the breeding value predictions module of TLR at the University of Pretoria.